Hello friends, today's topic of discussion is transcription in eukaryote. It is somewhat more complex as compared to prokaryotic transcription. This may be asked as LAQ in university examination as well as SAQ, and it contains MCQs for entrance examination. So this is an important topic for exam preparation, also entrance preparation. We'll discuss this transcription process with respect to these headings: definition, then RNA polymerase 1, 2 and 3, various factors, that is basal transcription factors, activators and co-regulators required for the transcription process in eukaryotes, and various steps, that is initiation, elongation and termination of transcription in eukaryotes. Okay, so what is the definition? As all of you know that transcription is process of synthesis of mRNA or process of synthesis of RNA from the DNA template. So this is DNA which is transcribed into RNA. This process is known as transcription. All of you know the terminologies, that is promoter region. Promoter means it is a region on the DNA molecule to which RNA polymerase can bind. Okay, this is known as promoter region. Then what do you mean by template strand? It is a strand of DNA which act as a template or which act as a mold for the synthesis of complementary RNA molecule in a specific and organized way, that is known as template DNA strand. What do you mean by coding strand? The opposite to the template DNA strand is known as coding strand. Why it is known as coding or sense strand? Because the sequence, nucleotide sequence in the coding DNA strand is exactly same as that of newly synthesized RNA molecule. But the RNA molecule will have uracil and the DNA coding strand will have the thymine. This is the only difference. Okay. Then what do you mean by upstream elements and downstream elements? So suppose this is a DNA strand and this is a coding strand of D. This is coding strand and this is template strand. So coding strand in 5' to 3' direction, and this one in 3' to 5' direction. This is known as template DNA strand from which RNA molecule will be synthesized. That is known as template strand. Okay, this element, this is a, suppose this one is +1 is a transcription start site. From this nucleotide transcription will start. Okay, this whole unit, it is known as transcription unit, and RNA synthesized from this transcription unit is known as primary transcript. Got it? So toward this side, left side of this transcription start site, it is known as upstream elements and it is represented by minus sign, while toward this side it is known as downstream elements. Downstream elements. Okay. Now what is promoter region? It is a region on the DNA molecule where RNA polymerase can bind. It is known as promoter region. So this is promoter region. This another region is known as promoter proximal region. And again there is sequences which helps in the regulation. They are known as distal promoter sequences or distal promoter region. This is promoter region, proximal promoter region, and this is distal promoter region which helps in the regulation of this transcription. Okay. Now RNA polymerase in eukaryote, it is of 3 types, 1, 2 and 3, and this differ from each other in sensitivity to alpha amanitin. Alpha amanitin, sensitivity to alpha amanitin. This is MCQ. Okay, so RNA polymerase 2, it is sensitive to the alpha amanitin, which is a toxin from the mushroom. So there are three types of RNA polymerase, that is RNA polymerase 1, 2 and RNA polymerase 3. So RNA polymerase 1, it is involved in the synthesis of ribosomal RNA, particularly 28S rRNA, 18S rRNA and 5.8S rRNA. RNA polymerase 2, it is involved in the synthesis of mRNA and small nuclear RNA. While RNA polymerase 3, it is involved in the synthesis of tRNA and 5S rRNA. So these are the three different types of RNA polymerase in eukaryotes. Okay, and this RNA polymerase 2, it is sensitive to alpha amanitin, which is a toxin from the mushroom phyllodes. RNA polymerase 1 is insensitive, while 3 is having intermediate sensitivity. Okay, so these are the three types of RNAs involved in the transcription process in eukaryotes. Now there are various factors, they are known as activators, then basal transcription factors and co-regulators. They are involved in the transcription process. Near about 50 proteins, they are involved in the transcription in eukaryotes. So number of proteins are involved in the transcription process in eukaryotes. So activators includes SP1 protein, ATF, CTF, etc. Basal transcription factor, it includes TATA binding protein and various transcription factor, that is transcription factor 2, A, 2B, 2F, 2E and 2H. These are the various basal transcription factors involved in the transcription process. And co-regulator, it includes the transcription factor 2D, which is composed of TATA binding protein plus number of near about 14 transcription associated factors. They act as a co-regulator in the transcription process. So these are the various factors involved in the eukaryotic transcription process. Okay, now we will see the different steps in the eukaryotic transcription. So initiation, elongation and termination, these are the three important steps in the transcription process. So in the initiation, there is formation of pre-initiation complex, there is formation of open complex, then there is formation of stretch of RNA and promoter clearance. These are the basic steps in the initiation of eukaryotic transcription, followed by elongation and termination. So we'll see one by one with diagrammatic representation. So this is DNA molecule. This is transcription start site, that is represented by +1. There is -25 to 35 region, that is upstream. There is one region that is known as Tata box. So -70 region, there is another region that is known as cat box. And again there is sequence that is known as GC reach region. Okay. So this is promoter region, this is proximal promoter region, okay, and this is also proximal promoter region. So these are the sequences, they are known as consensus sequences on the coding strand of the DNA. This is coding strand, this one is the template strand. Okay, this is upstream, this is downstream. This whole unit is known as transcription unit. Now number of factors are involved in the transcription and pre-initiation complex formation. So this mainly includes the sequence. First there is attachment of TATA binding protein followed by transcription factor 2D, then transcription factor A, then B, then F with polymerase, then E, and then H. E and then H. This is the sequence for attachment of various transcription factors to the promoter region. First, there is Tata binding protein. This protein will bind to the Tata box, that's why it is known as Tata binding protein. And to this protein, there is attachment of transcription factor 2D. So, to this protein, there is attachment of transcription factor 2D. Okay. Attachment of transcription factor 2D. What is the role of TBP here? It recognizes the Tata box. Okay, scan the DNA molecule, recognizes the Tata box. Then after attachment of TF to D, there is bending of DNA occurs. Near about -100 degree bending of DNA occurs. This is because of this TFT2D, and due to which all these factors they work in a coordinated manner due to this bending. Okay, so there is this is Tata binding protein and this is TFT transcription factor 2D. This is Tata binding protein. Okay, now the next factor A and B will stabilize this complex, and B help in to stabilize this complex. Then F, transcription factor F with polymerase attaches to this. This is polymerase with F. Okay, then transcription factor E and transcription factor H will attach. So what is the role of TBP? It helps in the recognition of Tata box. Transcription factor 2D will causes the bending of the DNA molecule. Then A and B will stabilizes this complex. F, transcription factor 2F, it brings the polymerase, RNA polymerase to this site, and then attachment of E occurs and H occurs. So H has number of function. It has helicase activity. It is responsible for unwinding of DNA molecule. Also, it activates the carboxyl terminal domain of the RNA polymerase. It phosphorylates the carboxyl terminal domain of this RNA polymerase and activates the RNA polymerase. Then third one is it also involved in the excision repair, nucleotide excision repair. So you will get MCQ on this part H, transcription factor 2H. Helicase activity, phosphorylation of carboxyl terminal domain of RNA polymerase and activation, and excision nucleotide, it is involved in the nucleotide excision repair. Okay, so these are this is known as formation of pre-initiation complex. Then there is opening of this DNA occurs. Opening occurs and synthesis of RNA will occur. So RNA will be synthesized. Okay, so stretch of RNA will be synthesized. After this, the RNA polymerase and after this, all these factors will dissociate, and RNA polymerase moves away from this promoter region, that is known as promoter clearance. So what are the steps in the initiation process? Formation of pre-initiation complex with the help of attachment of various factors to the promoter region. Then this closed complex turns into the open complex by unwinding of the DNA strand. Then synthesis of RNA molecule occurs, and after that the promoter clearance will occur. These all factors will dissociate from this region, promoter region, and the RNA polymerase moves physically from this region to the another region of the DNA molecule. So it will now, this is RNA polymerase with factor F. Only factor F and RNA polymerase will remain, all other factor will dissociate. Okay, this is the initiation of transcription. Now elongation will occur. In elongation, all of you know that there is formation of 3' 5' phosphodiester linkage. Polymerization means addition of nucleotides. Addition of nucleotides complementary to the DNA strand. Okay, only in RNA there will be uracil in place of thymine. Got it? So there is elongation and then termination. So we'll see the termination. There are intrinsic signals for the termination to proceed in the eukaryotes. These signals are, we'll see by the diagram. So this is DNA molecule. This is template strand. This is newly synthesized RNA. This is 5' end. So there are intrinsic signal, that is A, U, A, and CA. These are the intrinsic signals for the termination in eukaryotes. So the one protein, that is cleavage and polyadenylation specific factor. Cleavage and polyadenylation specific factor will attach to this site, that's this sequence, that is AAUAA, and cleavage stimulation factor will attach to the CA sequence, and they will recruit one factor that is known as cleavage factor. This cleavage factor has endonuclease activity, and this will bring about the dissociation of DNA RNA complex. This will help in the dissociation of DNA RNA complex. So this is termination in eukaryotes. So this is all about the eukaryotic transcription process. So keep watching. Thank you.